relationships with their symbionts. But we're trying to genotype all the symbionts right now, and as well as trying to figure out ways we can sequence their whole genomes to figure out how they're contributing to host metabolism. That experiment I just showed you, we have stable isotope analyses in progress right now. I was hoping we'd have the data by now to show it. But we're still trying to process that to figure out how stable isotopes change during the experiment. And we're going to use the stable isotope numbers to come up with a quantitative model of these costs and benefits and really try to do better projections of that. Okay. All right. So this is our idea of a qualitative model so far. We had a great review of all this work in Advances in Marine Biology last year. And our idea is that with our sponge host, we think they have some beneficial symbionts that are providing either carbon or nitrogen or chemical defenses back to the host. But we think the host has to be supplying something, but we just don't know exactly what yet, whether it's carbon or nitrogen or phosphorus or some other limiting nutrient, right? We know that there could be some growth inhibitors going on. Some sponges do make growth inhibitors, or they could just eat these things by phagocytosis, right? We also know that some of these symbionts are harmful. They can overgrow the sponge, block the canals. We've even seen damage to different skeletal structures in the sponge because of these symbionts. But we want to know, can all these things change based on environmental conditions or different amounts of nutrient cycling in an area or competition for resources or being colonized by some other symbiont, right? So that's kind of our model right now of how we're trying to investigate all of these interactions. Okay? But my bigger goal is to take all this information and then to map that onto our phylogeny of sponges, right? Because I told you a lot of these sponges that we started working with, we didn't really have names for, right? So that led me to become involved in this Porifera Tree of Life project, trying to map the evolution of all sponges. And I have a selfish reason for doing that. I'm going to take all these symbiont characters and map them onto that same phylogeny, right? So that leads us into this Porifera Tree of Life project. I'm just going to spend a little bit of time on, because I realize a lot of y'all might not want to look at picky taxonomic questions and everything for the rest of the day. But to give you an overview, in 2000, there's a great paper came out in Hydrobiologia that's Molecular Systematics of Sponges, right? And it's by Carol Borcellini. And it summarizes in just about 12 pages all of the DNA sequences known for sponges at that time, right? And when that came out, it was great, too, because it showed you that different labs do different things. And so this was her representation of all the sequences with 18S sequences now, or in 2000 in GenBank. And some labs have the whole sequence here, and some species have the whole sequence there. Some of these have just a really short thing. Sometimes that's shifted over, right, so they don't overlap much. And then some labs actually have 3 separate pieces, right, that are really short, and they don't overlap either, right? So one of the goals of the Tree of Life project at NSF for all these different groups of organisms is to fill in these data gaps, right, and fill in those data gaps. And it's a little bit low reward, right, because you're asking people to redo work that they already did, right, and nobody really wants to do that. So what we did in my lab is we contacted people that had data sets like this and said, well, mail us all your samples and we're going to fill it in, okay? And we're trying to get a complete phylogeny for the group. And our papers, our first big set of papers came out just this month. If y'all go to Integrative and Comparative Biology and look at this month's issue, you'll see there's 12 papers that are sponsored by Portal. Okay, and for the 18S sequences, this like I said, this is about the full amount they had in 2000. This is 40 different sequences here, and we published already, I think, about 780 that were in this last paper. Right, so we have a lot more that we can do as well. So just to refresh your memory about sponges, there's 4 different classes. Okay, so you got demosponges, hexactinellids, calcarea, and homoscleromorpha. And they all 4 have really different characteristics to establish those classes. So when you think of a sponge, it's like a bath sponge, or it has, say, fibers in it, or just, maybe a few spicules in it. That's usually a demosponge, right? If you've got a deep water glass sponge, that's these hexactinellids, okay? If you have a calcareous sponge, they're all in calcarea, and they have calcareous spicules in there. And then these homoscleromorpha, you might think they were part of the demosponges. They have little spicules like a demosponge would, but they actually have other features that are more like other metazoa, like having a basement membrane. They have gap junctions between the cells and things like that the rest of these classes don't have. Okay, so they have a lot more biochemical features that you might consider part of Cnidaria or something like that. Okay. When we look in the demosponges, then there's 4 big groups, and we were calling those just G1, G2, G3, G4, but that's not very descriptive for people. So in our papers that come out this month, there's actually names attached to the groups now. Okay? And we can think, how are these reflected with just the names of sponges that we know today? Well, most sponges that are described today are in this G4 group. Okay? And just a very few things are homoschleromorphs or even calcarean hexactinellids. Okay? But most things are in this G4. So that's still a big area of research for us. For Portal, what we decided to do is kind of trade off our sample effort and our sequencing efforts. So we sampled at the family level. There's 142 families of sponges to try to get some nuclear housekeeping genes, whole mitochondrial genomes, and 18S and 28S sequences. At the genus level, we dropped out all these housekeeping genes, and we just did 1 nuclear and 3 mitochondrial genes. And then for about 8,000 species, our target is to do 2 barcodes. We have a mitochondrial barcode and a nuclear barcode, and then to do morphology on all 8,000 of these. Okay, so that's where we're spending the bulk of our time in now. We've kind of completed these first 2 steps, and we're down in this lower run. Okay. In my lab, we really focused on 28S, and you have to remember I'm SpongeBob, right? So everything in my lab has SpongeBob on it, so it's an easy target, right? But, so we were trying to do this whole 28S RNA gene, and it's really difficult to sequence the whole thing all at once, or just to use PCR to amplify the whole thing at once. So it's about 3,500 bases long, and usually we couldn't get the whole piece. I mean, that's like maybe 10% of our samples we could. And instead, we break that into 2 long chunks. Sometimes that doesn't work, and we could do 2 chunks at the front, or just this barcode region we have. And we have a long flowchart that my lab had to use to try to get this to work, because we had a lot of things where we would get our DNA extraction. The extraction would look good, and you could never PCR anything from it. Okay, or we would have things where you get a sequence, and then, oh great, it's this polychaete that lives in the sponge, right? Or it's a copepod, and you're like, oh great. So you got to go back and start again. So we had a long loop of that sometimes. But I have to say special thanks to Cheryl and Megan, are 2 technicians that work in my lab, and then Lori Spicer is with our collaborators at University of Richmond, and they did a ton of the lab work for all of this. Okay. So once you get out the ribosomal genes, whether it's 18S or 28S, and so at Richmond and at UAB we worked on 28S, and the Smithsonian labs did the 18S. This is the 18S ribosomal secondary structure all folded up. And so when you're looking at these sequences and you want to try to analyze the phylogeny of these, you really have to take into account that secondary structure, right? So that means you got to do a lot of folding, and this is really computationally complex. So we had to work with a lot of different groups to try to figure out how we could get the computational resources together for that. And then what I'll show you today is that we had to limit our analyses to sequences here that are greater than 2,000 base pairs. Okay, that's how we get the optimal alignment for the software like MAFT is one that we use a lot for getting these. And then if we made trees, we used Raxamel and Phylobase. I'll show you a tree in a second. But the sequences that we used, these 327 sequences I'll show you just now, 65 of those were in GenBank when we started the project, right? And then 57 are from our collaborator, Christine Morrow, and she's a graduate student in Ireland at the National Museum of Northern Ireland. Okay, and this was great because when we were able to help her with her doctoral work and help her figure out parts of her project as well. About 72 of these species are from Panama, and then about 150 are from the National Cancer Institute. So that's, I mean, they were a really big source of all of our sampling. When we look at our 18S and 28S trees, this is just one to show you how congruent they are. And the bootstraps here are based on these kind of Bayesian analyses of these phylogenies. And you can see that we have these major groups of demosponges. So the keratosa are what you used to call orders Dendroceratida and Dictyoceratida. There's the G2 is this mixospongy, which is Varangida and Chondrosida put together now. We have 2 things kind of coming splicing out in this 18S phylogeny that we're not quite out in this 28S, and that's the distinction with your freshwater Haploslerida and what's known as your Dictyonellidae. Okay, so we're still trying to get a lot more genetic data from those 2 groups. They're not heavily sampled in here yet. Then for this G4 group, that's just now a mishmash, and it's called now the heteroscleromorpha, which means just that there's a lot of different kinds of spicules in those. Okay, that's how you get this heterosclero in there. But This is the orders like Agilacida and Hadromerida, Helichondrida, and so forth. All your marine Haploslerida are in one group together. And then the glass sponges stick out together. The Calcareous and the Homoscleromorphs are all really distinct. Okay. So if you want to get more interested in this, the special issue of Integrative and Comparative Biology delves into all these different clades a lot more. And you can drill down into each one of these. And we have basically a paper on every one of the major clades there. Okay. All right, so when you look at this big tree, though, you can see that it's really complicated just to get those 370 sequences aligned in sequence. And now I've got about like 3,000 more barcodes to staple into the giant tree, right? So we're trying to figure out how we're going to do it. And that's where this Avital project comes in. For Avital, there's 3 big components to it. One is the open tree, and I forgot to make them a slide on here. But if you go to Open Tree of Life, that's where you can see the giant tree of life that's coming together from researchers pulling together every published phylogeny. Okay, so the group with the open tree is really trying to pull out every animal tree that's in literature, every plant tree, and then staple those all together into this ginormous tree of life. Okay, we've also gotten them now working on microbial trees, right? And so it's going to be a really fantastic product. Right now you can go on there and take a look, and there's some ways you can interact with it, but they're going to be building a lot of new functionality in the next year. There's also a project called Next Generation Phenomics for the Tree of Life, and so I'm involved in that one. For that one, we already had a thesaurus of sponge morphology that we used to look at sponge spicules and use proper names when describing sponges. But we've converted that now into an ontology, and if you go to Google Code, if any of y'all are doing any programming with Google Code, you can just type in Porifera ontology or just do a Google search for this. You'll get to this page, and it's our ontology of how all these terms in the thesaurus are all related to each other. Okay? We're kind of using those terms now to power a new way of describing species so we can describe them a lot faster. And that work we're doing in conjunction with the phenotype RCN. So if any of you have work that you're doing where you're describing phenotypes and you're trying to figure out what's the best way to talk about a phenotype for one organism or another, this is a great resource. It's just phenotypercn.org. They've had classes every summer the past couple of years on how to build ontologies, how to really structure descriptions of species or features. And so this is really starting to get off the ground really well. The other group that I'm in, and I spend most of my time on, is this group called Arbor. So we're trying to do comparative analysis workflows for Tree of Life. So right now, if you want to do a big analysis, say, of some trait evolution in your favorite group, you've got to get a tree, you've got to get a table of traits, and then you've got to do a lot of analysis, right? Trying to figure out how to map those characters onto the tree. Right now, there's probably like 20 different steps of software you've got to use. So we're trying to pull all that together into a nice workflow. Most of it's going to be in R, but we have our own kind of standalone platform for it. But the idea here is that we'll be able to access remote data sets. So whether you're going to GBIF, that's your global, I got to remember, Biodiversity Information Facility. If y'all go to gbif.org, you can check that one out. But you also GenBank and all kinds of other things. Then you can do some computation on your desktop computer. But what we're really going to do is send it from your desktop to some web services that are housed on the National TerraGrid, National XSeed network. Okay, so you'll be able to do computation on these really high-speed machines, and it's going to come back to your local computer for your visualization steps. And then the key part is that we're going to have access to remote archives like Dryad, and there's a couple of new ones that are being developed by iPlant. If any of y'all are doing terrestrial plants, you go to iplant.org, you can see their archives, right? But we're going to have good ways you can store your data and cite it. Okay, and so the idea here is to really make this a seamless process so that you can grab data from a lot of different sources around the world, you can process it really quickly, and then you can share your results really fast. Okay? And then we have 3 case studies that we're working on. So one of these is, we call it this evolutionary process of spatial diversification, and we're really looking at the evolution of an ecological niche, right? And how can you use ecological niche models not just to predict the distribution of the species, but then to map them onto a phylogeny and look at the evolution of that niche. Okay? And so part of that is kind of diagrammed here, our workflow for that, where you're combining data from GBIF, the Climate Data Service, and Treebase here to integrate all that data, build a map. This is looking at flora of Mexico here on this map, and then build that into a phylogenetic tree, say, of those plants, and then upload all your results. I'm kind of more focused on the second one, as you might imagine, the evolution of symbiotic communities. So I'm kind of directing our project in that group. But we're not just looking at sponges and microbes. We're looking at anything that has a symbiont, right? So we're really trying to track huge data sets and get through a lot of those right now. The third one we have is this evolution of complex interactions, where we're trying to look at evolution of food webs and evolution of community structures. So that also feeds into the symbiotic communities, because we're trying to figure out communities where we've got really good documentation of all the processes and interchanges going on, and then can we map the evolution of that over a large part of the tree of life. Okay. So here's some take-home messages for y'all for this today. So I want y'all to figure out that sponges do provide a really model system for looking at the evolution of microbes, communities, right? And we know that we can put these communities onto our sponge phylogeny now, right? So that's one of our major goals, is how do we look at the evolution of that microbial community through the host. We know we can manipulate these to test hypotheses about cost and benefits. We can use our shading experiments to do that, and now we've got some really great tracers we can use with our stable isotopes of carbon and nitrogen. With our Portal project, we know now that our sponges are this ancient monophyletic lineage, right? We've got deep diversification events in there, and we're really trying now to puzzle out the structure of different genera and families within each of these big major clades of the Porifera. As part of the Porifera too, we've got these huge samples collected, right? This huge set of samples. So we're partnering with Earth Microbiome Project right now. We've sent a bunch of samples to them so that they can actually survey the microbiomes inside of those sponges, right? So that's where we're heading to next, is to try to look at this huge diversity of sponges, but also look at the huge diversity of bacteria in them simultaneously, right? And then to handle all that data that we know we're generating, that's why the Arbor project is really important to me. We really need this big framework for testing evolutionary hypotheses with all the phylogenies that we're going to have from both the microbes and the hosts. Okay, and then I liked what Maya said earlier about having a royal we for the thing, right? But this is my lab listed up here, and all these collaborators that have been part of our Tree of Life. And this is probably just like 1/10 of the people that have helped out with sending samples from around the world and sending us sponges. Okay? And I'll end with this one. This is a new sponge that was just described last year by Welton Lee. This is a chondrocladia that lives off of the coast of California in Monterey Canyon. Right? But this doesn't look like a sponge at all to you, like what you would think of as a sponge, right? This actually doesn't filter bacteria out of the water. It's got little hooks all over it, like little Velcro. And when a copepod or a crustacean hits that, then the sponge cells grow over it and digest it, right? So it's more like a carnivorous sponge, right? So this one's really wild, but it's got this pentaradial symmetry and everything. It's just amazing. So this is one thing to keep in mind with this world of sponges and these microbial symbionts, there's always something new to discover, right? And there's brand new stuff out there every day that we can go look for. And so that's what I find really exciting about all this stuff. So, all right, that's all I've got. I'll be happy to answer any questions for you today. Okay? Sure.
Thank you very much for the talk, Dr. I have a quick question about the shading experiments. Have you given thought to what the cyanobacteria are preventing from moving into the organism and killing the cyanobacteria? You're actually opening up a niche for possibly a harmful bacteria.
Right, and so that's a great idea. We haven't gone out, and one thing we're trying to do is, I've got a student that has that same idea right now, where he's trying to do the shading experiments again, but then analyze the whole microbial community structure before and after the shading, right? To try to see that, like, what else have you altered, right? And so that's a great question. So we're still trying to figure that out, but it ends up being pretty tough because we can track the cyanobacteria really quickly. We can just make a histological slice and look in the fluorescent microscope and see them, right? All these lots of like alpha proteobacteria, gamma proteas. It's not that simple, and so that's what we got to really try to figure out. Yeah, sure.
So I'm really interested in this Arbor project. That is so cool, making some use out of all those phylogenetic trees that we always read. Is this eventually going to be available for public use?
So we have some test stuff now. If you're feeling really brave and things are and troubleshooting a little bit, there on GitHub we have a whole site. So if you went to it's arborworkflows.com, you can I can actually pull that slide back up. It'll be.
We can break it. within days, I
Oh,
guarantee.
gosh.
Oh yeah,
You
y'all
have
can
a
probably
version
break
by
it. There it
Monday.
is. So, this one here, if you go to arborworkflows.com, you can download some test stuff. There's a site that you have to go to. It says developers or whatever, and then it'll send you to GitHub, and you can just download some stuff to try it. Yeah, it's great. Yeah.
Well, this slide intrigued me. Who do you have working on the 4M symbiont communities?
So we don't. So would you like to work on them? Right, because that's what we need. Right. So right now, what we're trying to do is build the way we can test it, right? And then we're trying to recruit people to help with data, right? So here you go. Here's the shot. So, but yeah, that's that's what we want to try to do, is to try to build a platform and then make it super easy for you to test your own hypotheses and get in there. So we're thinking by January we'll probably have some stuff that we can release, and then we're going to have workshops to try to bring people in and say, okay, here's how you can use this to get to your question, and that's where we have to figure out what other features people want and things like that to visualize, right? Yeah.
Well, the interesting thing is the 4Ms have the longest fossil record.
Right. And so that would be one we can, because we could calibrate with that too. Most of the team, I can say, is so working on our plant kind of modeling and some bird modeling is Jorge Soberon. He's at University of Kansas, and then Luke Harmon works on more herpetology type things up in Idaho, and Chelsea Specht is more plant-based at Berkeley. And so I'm the more microbial person and marine invertebrate person on the team. So we're trying to figure out how we can address a lot of different research communities, though, because they're coming up with ideas from, say, the plant project world that I hadn't even thought of, and things like that yet. So yeah.
Yeah, I didn't quite hear the first question, but I was worried about, are there microbes that are attacking and killing the sponges? Because I know we have
right.
The big barrel sponges are dying on
right.
Joe Lopez.
And Joe Lopez working with that as well, right? So we know that
sponge bites in the past wiped them out all over the
That's right. So we know that we have different microbes that attack the sponges in different ways. So there are a couple studies there's one that's been cultured. I think it's a gamma proteobacterium that goes into the spongin fibers and actually eats them, right, and dissolves around them, and that's how that's like the substrate that the bacterium grows best on, right? So there are some definite pathogens that attack. I didn't show you a story of these, the Aplicina red band syndrome, but I've been working on that for a couple years. It looks like a leptolingbia that attacks the sponge. We see lesions with a red band around them, and if you open up that red band under the microscope, it's just packed with cyanobacteria, right? But we don't know if that's a secondary infection, right, or what's actually causing the primary infection. We've actually cultured a leptolingbia, but we couldn't create the infection that way.
I seem to recall that some sponges were bleaching back then when the corals were bleaching.
Right. And so some will, and Joe Pollock has a great study of cyclical bleaching with the big barrel Zetspongia muta. Okay, so they'll actually do that in a cyclic pattern, and we're really interested to know if for the Zetspongia muta, there has been a couple shading experiments where there's no impact on the sponge. So we're really wondering whether those are commensal cyanobacteria in there, or are they somehow harmful, and so the bleaching can be a way for the sponge to get rid of them, right? And we're still trying to figure that out. That's a really big open question, right? Yeah.
But there are a lot of other bacteria species in there.
Right.
So when you shade and
Right.
with the
It's only right.
you have all sorts of positive and negative.
Right, and so that's what we're trying to get into now, is trying to take our samples and look at the whole microbial community before and after the shading stress. But that's just now what we're getting to, right? So we started out with just focusing on the cyanos, just to see something we could measure that was functional and kind of as a pilot, right? And now that we know we can look better at the whole community with these new sequencing methods, and we're using like Illumina sequencing and stuff, what we really want to try to disentangle that now. Yeah, right. It would really be good, right. Yeah.
So for your have you done many colonization experiments, or you've only looked at the cyanobacteria?
Okay, we haven't done many any work with like how something colonizes the sponge substrate, right? So we haven't done anything with that yet.
Okay, so different sponge species in the same body of water that all have
Right.
what you hypothesize
Right.
its own specific.
Right, and so with those, let's see, I can back up with all the ones that we've been studying for looking at specificity so far. Those aren't manipulative, right? So we're not manipulating the interaction at all. It's all survey based so far, right? And that's where we're trying to figure out, can we get some of these into culture and then do these kind of transfection? But we haven't gotten most of these bacteria ever cultured, so yeah.
But you've done, you know what's in each of these.
Right, but that's what we're trying to do now, is figure out what they are, right? And yeah, in the back you had one, yeah.
Do you think the symbiosis could be providing any benefits to the larva?
Oh, yeah, we've been wondering about that, especially these ones that swim around with cyanobacteria in there. We're wondering, are they actually photosynthesizing while they're able to swim in the water column? That one that I showed y'all that's really packed, the interesting thing is that its larvae don't last very long. It's like less than a day before they'll settle. So, but we have some that have a lower level of cyanobacteria inside them, and that's ones I'm really curious about, because they'll stay in the water column for 4 or 5 days and things. So that could maybe be a bigger benefit for them. Yeah. And you had a question too, right?
Sorry to be greedy.
Yeah.
Hasn't nature given you the best shading experiment ever in depth?
Right. So we've done some depth transplants, some reciprocal transplants with depth, but those, it's curious because a lot of other factors are usually changing simultaneously. So that's why I don't know, we haven't seen the best results with those yet. I've got a student that started a long-term depth transplant in Panama this year. And so that's where we're trying to see how well will that work or things, right?
Just if the organism, if you believe that the organism requires the cyanobacterium to live, has that organism been found at deeper depths where that cyanobacterium can't go? Right.
And so
transplanting, just yes or no.
Right, yeah. I see what you're saying, yeah. And so we did a big survey like that. We stratified actually the survey I showed y'all with abundance on the reef. We had different depths in there, and we didn't see any impact in that study. If we're looking at these oscillatoria, they are really specific to where those species live, right? And most of them have like a pretty narrow depth range. Some of them have a wider one in the Indo-Pacific because there's light is penetrating much further down, right, in different areas. Where we work in Panama, it's really interesting because the light's not getting very far down, like maybe getting down 40 feet or something, but it's really murky and stuff. So we wonder about that a lot, like that light habitat, especially that light gradient. So yeah, definitely. So yeah, oh yeah, behind.
I was just curious. When you're going to do the microbial community shift studies, are you also considering looking at the metatranscriptomics?
Right, that would be great. So no one in my lab has done transcriptome stuff yet, so that would be a really great thing to add. So if anybody's interested in doing some transcriptomes with us, we haven't even tried that yet, but that would be really good to do. Right. Yeah.
Can you go back to the the stable isotope, your Freeman's work?
Oh yeah. Okay. I can come back. That's where your brain's at?
Because it doesn't make sense.
Let's go back. How far do we have to go?
You want me to jump on the computer?
Oh, maybe I could. No, it'll be good. There we go. Well, no, it's slower. Sorry. Okay, there we go. Okay.
Okay. The red is the shaded, right?
Right.
Okay. If they are photosynthesizing more slowly, they are able, they're less dependent upon the okay, I misread this earlier.
Okay.
I was wondering if they were just photosynthesizing more slowly, and that had to do with the change in the
Like
in the isotope.
this downshift here, And so that's one thing we're really intrigued about, because I'm not sure how that's happening, right? And that's why we also aren't sure that the genotypes of the symbionts didn't change during the experiment, right? So we're trying to come up with some really good ways to actually look at the genotype of these, and actually maybe if we can do some whole genome sequencing, we could figure out what the photosynthetic machinery is in there and things like that. It is really unusual. We still haven't gotten a good explanation. So.
Can you measure the DOC?
Well, that's one thing we were wondering, if they could be switching to the DOC to come in and out. But we didn't measure it during the experiment, right? We do have a way now, though, we can do these in light dark bottles, right? And we've been doing incubations in light and dark bottles and spiking that with the isotopes of interest, right? And then we can kind of track it through time that way. And that's turning out to be really a neat way to go, so we can try to measure photosynthetic rate and the isotopes at the same time, right? But we don't really have I don't really have data I'm confident in yet to show you with that. So.
Can you go to actually the next couple slides? Your
like this? Yeah.
Are you familiar with Diane Stoker's work?
Yeah. So we're trying to figure out just a good way we can model this, and then what we want to do is take the actual rates of transfer that we can measure with our incubation experiments, right? And then try to add those in. So what's the maximum amount of, say, carbon be transferred in, like, an hour or 2 hours, something like that, and then see what the respiration rates of the sponge are and all that kind of stuff, and try to put it all together. But we're still in this thinking about what are all the things we need to measure to build the model right now.
Maybe we'll just take one more question, and then Bob will be around, and he'll be around for TGIF. You can catch him after.
Yeah, sure.
Is your Syngicoccus actually spherical, as it looks like from that TM, or is that a latitudinal cross-section?
It's a little oblong, right? Because these are all like sections when we look at it, and you can see sometimes it's a little more oval, and sometimes it's perfectly round, and so it just depends on where the section is and stuff, right?
All right. Thanks, Bob.
Okay. All right.